Thank you very much. For the students who could not attend the lesson of 31st March, I will give you a very rapid outline of the lesson. And please ask your colleagues to get the full notes or explanations in case something is unclear. And also you can write me an email with a specific question. Not, don't write, I didn't understand anything, because that, of course, is not useful. Anyway, let's get started. So the topic in this slide is showing you how many approvals for drugs have been done in the years from 1998 to 2018. And the topic of this slide is simply to show you that we have a very dynamic biotechnology movement, biotechnology industry, which is able to give a lot of compounds. And as you can see, the ratio of the red histogram, which is describing a biological license application, which means biotechnology, has steadily increased in percentage compared to the classic chemical drugs, and it's still on the rise. I've shown to you in class that there is a huge biotechnology market size with a predicted increase along the years, steady, with almost 60% increase in 6 years from 2024 to 2030. This is only for the American market. We have similar figures for the other very well developed market like Europe, slightly lower for the Japanese market. More or less in the Western countries, there is a lot of money invested in biotechnology. And in this lesson, I would like to show you why this type of investment arrived, showing you a successful story, and therefore also what some biology which is behind. In this slide, you see, referring to the 2018 year, that many of the compounds that have been licensed, many of the biotechnological driven drugs are represented by monoclonal antibodies that are the large majority. But also we have recombinant proteins which have been marketed like hemophilia A for hemophilia, and some nucleotide, be that interference RNA or antisense RNA. And obviously, again, this gives you an idea about the number of products, gives you an idea about how much this world of biotechnology is developed. About diseases, you may see that if we classify the type of drugs for diseases, we do see that a large majority, a large part is represented by oncology products, cancer. After cancer, we do see a lot of brain and nervous system related drugs, which are referring, of course, to mainly neurodegeneration. We do see investment in metabolic diseases, the liver, and other areas. So what is absent, as you can see, are rare diseases. For rare diseases, we don't have much of biotechnology products because there is not a big investment by companies, because the number of people that are affected by a single disease, by definition of a rare disease, are very few, and therefore the pharma industry does not invest in a market in which, unfortunately, patients are there, but they are too few to make the investment profitable. So, if we then classify the drugs that have been put on the market by the dimension of the company, we have a very interesting observation that big pharma companies, classical pharma companies like, for instance, Pfizer or big biotech, are representing only a limited slice of the market, We have a very high number of small biotech companies which are developed on a single idea, or regional or specialty companies devoted to a single disease that are also very aggressive on the market. And as I said during the lesson, perhaps you also will be one of the persons who will found his own company or her own company. You see a number one that I didn't explain during the lesson is a non-profit and go drug. This case refers to rare diseases where sometimes you have some kind of crowdfunding like mechanism, or granting agencies that are able to put together efforts and sponsor development of drugs for rare disease. So non-profit, non-government organization. And well, some examples of compounds are there. During the lesson, I made a big story on immunotherapy. Immunotherapy has been a revolution in cancer therapy, has been a change of paradigm because we have made a change from treating cancer through chemical drugs which are able to kill proliferating cells. By immunotherapy, we are able to treat cancer by using the properties of the immune system, by increasing the capability of the immune system to kill cancer cells. And the founder of this field is being considered in a way William Coley. William Coley was a surgeon in the States that at the beginning of last century was noting that some patients who underwent surgery for cancer and that had mild infections were able to recover from cancer. So the fact that infections were able to help the recovery of cancer patients led to the idea that infections could be used as a cure for cancer. The logic was not very well understood, but this was the empirical conclusion. And he started to treat patients with some kind of controlled infection, but without success. However, what is important is that the connection between an infection and curing cancer is true for some very rare cases, but still it is true, leading to the idea that somehow fighting an infection could be similar to fighting a cancer cells. Paul Ehrlich, who won the Nobel Prize a few months later, was the first one to describe the idea of antibodies and antigens, and Eli Mekshinoff was able to describe the concept of phagocytosis, so the fact that during an infection, bacterial cells are phagocyted. For those of you who are curious, these are key individuals who have been important for the history of medicine and the history of infection medicine. Paul Ehrlich is very famous because he was able to understand, even without seeing, the existence in the body of antitoxin, which were able to fight bacteria. And now the history is very clear. We have in the body what we call antibodies. Antibodies are able to recognize another molecule that we generally call antigen. And the binding of an antibody to the antigen is the key to immunological defense. So, trying to put in a context the fact that some infections were leading to cancer regression, scientists were then starting to study the immune system, and one of the many interesting things, many important discoveries that were made along the years are described in this slide. First, scientists discovered that T cells can be inside tumors, and that inside a tumor T cells can detect tumor-associated epitopes. So that is to say, antigens, tumor antigens. I explained during the lesson that these tumor antigens are represented by proteins that a cancer cell is expressing because due to genetic mutations, this protein is recognized by the immune system as a kind of non-self protein, and therefore a protein that is like the protein of a virus, a protein that classifies foreign cells. So by understanding that T cells were recognizing tumor-associated epitopes, scientists tried to boost the T cell response within tumor by using vaccination. And in a few words, they were using cancer cells to vaccine patients in the hope that the immune system was now recognizing the cancer as a virus or something similar as bacteria and fighting therefore cancer cells. But this was not leading to results, and there were very few clinical responses, and it was actually due to a poor understanding of the process of immunization. So when scientists discovered that during infections you had a production of cytokines such as interferon, and these cytokines are able to boost T cell response, they try to give to patients interferon or IL-2 and these drugs, and in the hope to stimulate the immune system to kill cancer cells. But also this was not working, and therefore there was the need to do something else. However, in the last 30 years, from the cloning of a gene, which is known as the CTLA4 gene, we had the first successes in this strategy, and the story I will tell you is how this success has been achieved. We will start from the Onjo Nobel Lecture of 2018, in which it started from common knowledge. The common knowledge is that if you immunize a person or a mouse, also or human, with an antigen, so you inject the antigen under the skin, and this leads to the production of antibodies. The response in green first is low affinity antibodies, IgM, and then a few days later, by the switch in the mutation, hypermutation, we start the production of high affinity antibodies like immunoglobulin type G, and we have a peak around, as you see, 10-12 days after the primary immunization. However, if you then wait around 28-30 days and you make a secondary immunization, you will see that here, first we have a more rapid eye-specific response, the immunoglobulin type G response, which is much higher. This is a 10 log scale, so it's much more than what it looks like, because we go from 50 as a response to more than to around 5,000, so 104 more response, and also faster response. So Onjo was trying to understand which were the mechanisms, not that they were responsible for the primary immune response, but for blunting the response between immunization. So in other terms, the idea is why the curve is like this, it doesn't continue like this. So somehow we must have a repression of the immunity program. Cut short a long story, he identified a single gene which was known as PD-1, and PD-1 was found to be a negative regulator of the immune response. By knocking out PD-1 in mice, you will see here that mice without PD-1 has a dilated heart, much bigger than the normal heart. They develop diabetes, they develop infection in the not infection, but inflammation, sorry, in the heart, which is known as myocarditis. This is normal heart with the fibers, and this is the heart with inflammation, with T cells which are entering in the heart. That's the reason it gets bigger. In other terms, he discovered that PD-1 is a negative regulator of the immune response, indicating for the first time that maybe when one of the reasons by which the immune response was blocked during cancer was not really the absence of antigens or the absence of antibodies, but the presence of suppression molecules which were able to suppress the immune response. He formally proved that. He made an anti-PD-1 antibody, and he used this anti-PD-1 antibody to treat a mouse model of melanoma, the B16 mouse melanoma. In the mouse which is not treated, as you can see, the liver becomes black because melanoma cells are metastatizing to the liver and they occupy the space. If you treat this mouse with anti-PD-1 antibodies, you can see that you have a strong reduction of the black staining, implying that cancer cells are now excluded or are killed when they go to the liver. So this is the beginning of the so-called immunotherapy of cancer. This is basically bringing back to the study of Cole, in which now we can try to think that the immune system can recognize cancer cells as an infection, but somehow the cancer is able to inhibit the immune response. And probably in some patients, during the surgery, the infection was able to re-stimulate the immune system in a mechanism that, of course, cannot be simplified in a lesson, but highly variegated depending on the tumor and on the patient. So these studies lead to an idea that the immune response is a balance between too much suppression, so you don't have immune response because you have proteins like PD-1 that block the immune response, or in alternative, you have increased immunity, increased immune response, and molecules such as PD-1 act shifting the equilibrium toward increased immunity. And to cut short a long story, again, nowadays we have several antibodies which have been commercialized that are able to target PD-1 and are used in clinical therapy for cancer. PD-1 is also binding, you see, antibodies against PD-L1, and there are also other antibodies that are used to treat cancer cells, and the reason is the following. In the nowadays model, we think that T cells, which are recognizing normally cancer cells or viral infected cells and kill them by stimulation from antigen. They have PD-1 as a repressor of the immune response, and tumor cells do have a protein which is known as PD-L1. PD-L1 binds PD-1, activates PD-1 signaling, and PD-1 signaling blocks T-cell receptor response. If you make an antibody against PD-L1 or you make an antibody against PD-1, you block the interaction between PD-L1 and PD-1. Now, blocking the interaction of PD-L1 with PD-1, these cells can be stimulated through the T-cell receptor, and the T-cells can kill tumor cells. Of course, this is a simplification, but more or less, this is the way it works. Another scientist, his name was Allison, was working on a related protein, which was known as CTLA-4. He actually made a knockout for CTLA-4, and he found that the mice that had the CTLA-4 knockout were smaller than the wild-type mice. And as I explained during the lesson, he actually showed that these smaller sites was due to increased autoimmunity. In other terms, if you shift the balance from immunosuppression to immune excitation, one of the main targets is of course the intestine, where we have a very active suppression mechanism due to the fact that bacteria must be kept in check. And the probiotic flora has to be kept has to be controlled. So by knocking down CTLA-4, we have increased autoimmunity, and one of the phenotype is basically the reduction of the size of mice that are unable to properly feed. However, Allison was showing that this was due to the immune system, because by knocking out also CD28 gene, and so this would be more or less the story. So when you have the T cell receptor of the T cells, which is stimulated by the antigen, you also require that CD28 is stimulated by antigen-presenting cells. So the co-stimulation of CD28 is necessary for having an efficient TCR stimulation. So what Allison was doing was simply to knock out also CD28. So by knocking out CD28 and CTLA-4, the double knockout recovered the sites, indicating that CTLA-4 was acting through the T cell receptor signal. And nowadays, this is the mechanism. You have the TCR here. You have the TCR receptor, which is stimulated by antigen-presenting cells, and this T cell is stimulated. But then, if you have CTLA-4 expression, you have inhibition of the proliferation. And the idea again was to make antibodies against CTLA-4. By making antibodies against CTLA-4, you block CTLA-4 activity. Now the T cells can be stimulated by antigen-presenting cells, because there is no more the inhibitory action of CTLA-4. This is a very simplified story. If you look at CTLA-4, you will see that it's organized, is expressed mainly in a subtype of T cells, which are T regulatory cells. And I will just give you some information about it later on. But the CTLA-4 acts again to block immune responses. So T cell activation can be obtained by the TCR activation and the CD28B7 activation. If CTLA-4 is engaged with the B7, as you can see, you have T cell inhibition. If you have an antibody against CTLA-4, then CTLA-4 is occupied by the antibody, and the T cells can act through CD28B7 normally and is activated. And nowadays, by using antibodies that are using anti-CTLA-4 or combinations of antibodies that are using CTLA-4 and anti-PD-L1 and PD-1, lead to very good survival in patients that are treated with immunotherapy. Conclusions of this part is that we have seen that the immune system has inhibitory molecules that are, for instance, CTLA-4 and PD-1, and that by making antibodies that are directed against CTLA-4 and PD-1, we block the suppression activity of the immune system, and we can get a response, a reactivation of T cells that recognize tumor cells. So, and this is the logical conclusion of the story that started with William Coley. Probably patients who were infected in the William Coley surgery were the patients who had reactivation of the immune system due to inhibition of either CTLA-4 and PD-1 by the infection, and the infection was probably stimulating the immune system, and this led to a complete reshaping of the gene expression in these patients. Of course, we don't have proof because the samples of these patients are not anymore existing, but it's certainly, it's almost certain that that was the mechanism. To summarize, PD-L1 PD-1 interaction inhibits T cell activation and attenuates effector function, and then blocking PD-L1 PD-1 restores or prevents loss of T effector function. And therefore, by using this antibody, T cells are able to kill cells. Overall, or more generally, what you've seen is just the tip of the iceberg, and the tip of the iceberg because we have to think about a very complicated pattern of immune suppression and of immune activation. So during the tumor development, for instance, tumor cells acquire expression of PD-L1, and by the acquiring PD-L1 expression, these tumor cells can indeed induce tumor suppression. And multiple mechanisms are likely to exist in this case. Example of this important type of approach is shown by original images by which a patient was having a terminal melanoma, where you can see the metastasis in the skin, known as cauliflower metastasis for its appearance similar to a cauliflower. And in the NMR, you can see, in the tomography, you can see the tumor mass here, and also internal metastases that are scattered in this part of the liver. A single injection of the antibody that reactivates the immune system leads to the very site-specific elimination of the metastasis. You see you have a hole instead of the metastasis, also similar in tomography, but you also see that not only this metastasis has been eliminated, but also inner tumors masses, like here. Here you really have in place of the tumor, now you have killing. Cells have been killed, and therefore this was a revolution in cancer therapy. Typical a very successful example of biotechnology. This is a Kaplan-Meier curve showing that combination antibodies, so antibodies that are targeting more than one checkpoint, immune checkpoints, have a better success than targeting a single checkpoint, of course. Other example here, overall survival, which increases dramatically in the presence of this type of drugs. However, this type of therapy is working only in a small subset of patients, 10 to 30%, and we have obvious reasons for that. And the obvious reasons are two. Of the two reasons is that the more a tumor is mutated, and the more it's possible that the immune cells recognize the tumor like an infected cell, and therefore eliminates the tumor cell. In other terms, as I said during the lesson, if you have melanoma, UV light are causing a lot of DNA damage, and these tumors, together with lung cancer of smokers, have a lot of mutations, and these mutations are acting as they look like to the immune system like non-self cells which have not been, which are like infected in a way, and therefore the immune system, if it's relieved from the tumor suppression mechanism of PD-L1 or CTLA-4, can easily attack tumor cells with high rate of mutation. And somatic mutation frequency, therefore, is a good proxy for mutation frequency is a good proxy for the immune response in tumors. On the contrary, if you have tumors which have very, very low mutation rate because the primary oncogene that are mutated are sufficient to cause cancer, you don't have we don't have a carcinogenic drug compound like smoke or UV light or whatever, those tumors that have low mutation rates are not able to respond well to immunotherapy. And this shows you the pathology that you can try to also predict the immune response by looking at PD-L1 expression in tumor cells. If a tumor cell expresses PD-L1 and the T cells have PD-1, then you know that giving the antibodies against PD-L1 and PD-1 may work. But the other reason for failure is the most common side effect of immunotherapy. As I told you, the immune system is acting balancing the increased immunity toward suppression, and therefore you can imagine that by shifting the balance to immunity, by giving anti-PD-L1 or anti-PD-1 or anti-CTLA-4, you also increase autoimmunity. So the T cells are now starting to to fight normal cells. So the most common side effects of immunotherapy is represented by autoimmunity. This said, there are several drugs on the market continuously developed. You can see them here just for a few of them. Each company is trying to develop its own antibody. Which is probably giving you an idea about how much is dynamic the field. As I said several times, we are in a tremendous moment in biology and biotechnology. The timeline of these discoveries is shown here, and I made a second example of immunotherapy by describing the CAR-TS strategy. In the CAR-TS strategy, what we do is to make a step forward, and now we take T cells from our patients and we engineer them in order to make them able to attack tumor cells independently from tumor suppression or other type of mechanism. So the idea is that once we know that the T cell is a T cell receptor, which can be activated, we develop now a chimeric receptor which is engineered, and this receptor which is engineered now will recognize both the tumor antigen, but at the same time should have molecules which allow the direct stimulation of the T cell response. In other terms, you don't require anymore many components in different cells, but you try to build a T cell, a CAR T cells, which is able to do everything by itself. And the name CARs is chimeric antigen receptor, because they are made of a chimera in which you have an artificial T cell receptor that recognize the antigen of the tumor cells independently of the mutation. Of course, in this case, you make it by engineering. You want to recognize whatever antigen it is. Doesn't have to be mutated, but you make an artificial T cell receptor and you put it chimeric with the molecule that is the T cell activating function into a single receptor. And in this case, you obtain in a way, you obtain a super T cells, a kind of a superhero T cells that go around in the body and basically kills everything it finds, which is recognized by the TCR. Obviously, you see here the story is more complex. You have many type of strategies that we are trying to develop with different type of dynamic receptor. You have different generations of biotechnological products, and also we have a suicide switch, because the main problem of these cells is that when they start to attack tumor cells, if the antigen is also expressed in normal cells, these CAR T cells would also kill the normal cells. So in order to prevent that, we engineer cells with a suicide switch. That if CAR T cells become too dangerous, you give a small molecule and they all die. And you can search internet and the scientific literature. There are a lot of things that are described in journals. Last but not least, I was actually telling you that one next frontier is trying to unleash the anti-tumor immune response through a more selective inhibition of the suppression phenotype. And all this starts from the observation that the suppression is driven by a single type of cells, which are the so-called CD4 Treg cells. And these CD4 Treg cells are actually in different tissues. So, in other terms, we have Treg cells in lung, Treg cells in the intestine, Treg cells in the cancer. They are all making suppression, but since they are enriched in specific tissues, we think that they have specific mechanisms to go in that specific tissue. Very clear law. Okay. And therefore, the idea is now not to act on general checkpoint inhibitors such as PD-1, PD-1L, PD-1L1 or CTLA-4, but to act on specific checkpoint inhibitors of the T regulatory cells, and not only that, but also tissue-specific Tregs. How this is done? It is simply done by, for instance, purifying Tregs from different organs, sequence the genome, the RNA of specific Tregs, and then trying to find the specific proteins which are expressed only in T regulatory that are, for instance, the tumor. So expression is made by several ways, so we don't have to go into details here. And by doing this, you may find markers for T regulatories that are only in tumor cells, and I made an example of this marker that is LANE. LANE is a Treg protein which is expressed only in T regulatory cells which are infiltrating tumors, and in principle, if now you make antibodies against these specific proteins, Treg specific proteins, then you are basically killing the suppression activity in the tumor, but since that type of Treg is not also in the lung, but only the tumor, you just have, in theory, kill the suppression state in the tumor, but you don't have any damage in other organs. Of course, this has to be demonstrated in the future. With this, okay, thank you very much for listening. This is a short version of the lesson that I did March 31st. Please see your colleagues for having further information, and thank you very much for listening. Have a nice weekend and see you at the examination.